research organization for social sciences and humanities, research organization for archaeology, language, and literature, research organization for health, research organization for nanotechnology and material, research organization for agriculture and food, research organization for governance, economy, and community welfare. BRIN catalyzing research and innovation to develop an advanced scientific ecosystem in Indonesia with two main targets. BRIN realizes the tremendous potential of the nation's youth in the field of science and technology. Thus, various research and innovation policies are being formulated to develop and cultivate human resources with high competence and aptitude in science and technology. Creating many excellent programs is a form of BRIN's commitment to researchers, innovators, and the general population from various disciplines across the nation. The research and innovation ecosystem quality improvement in Indonesia is also realized through the collaboration of various ministries, institutions, domestic industries, and regional governments, to name a few, by forming the Regional Research and Innovation Agency, or RIDA. Moreover, cooperation with various foreign research institutions continue to be developed through active participation as member of various organizations and international forums. BRIN organizes research into a more effective and efficient process by integrating human resources, infrastructure, and budget from various research and development institutions. Oriented by a national research priority, BRIN routinely enacts comprehensive evaluations from various research and development institutions. This effort is supported through the continued management and improvement of various research and innovation facilities located across 17 regions of Indonesia, dedicated towards the development of science and technology. BRIN continues to improve our contribution to research and innovation towards national development by developing and revitalizing 4 areas of science and technology, or KST. An integrated area consisting of research laboratory facilities, testing laboratories, and industrial facilities for both startups and SMEs, tenant office rooms, co-working space, meeting rooms, gallery rooms, and other supporting facilities. BRIN is here to manifest the future of a golden Indonesia 2045 by consolidating science and technology resources and creating a resource ecosystem and research-based economical foundation. Transforming Indonesia as a global reference in the development and application of science and technology practices and the good of humankind. Nature is the source of inspiration. Knowledge is the foundation, and to be put in praxis for the sake of the nation. Research is our strength to overcome any limitations. Innovation is the guiding vision that drives us towards the future. We cannot face this alone. We must stand together to make our vision a reality. growing strong upon the land of Nusantara, a magical place filled with miracle and wonder. A land spanning 2 continents and 2 oceans, rich in culture and biodiversity. We and Indonesia are the necessary combination to realize the grand visions of the future. Like an orchestra, BRIN harmonizes research, development, assessment, application, and also synergies, invention, and innovation in Indonesia. We created an ecosystem consisting of 7 deputies, 12 research organizations, and 85 research centers dispersed throughout Indonesia. Research Organization for Aeronautics and Space, Research Organization for Nuclear Energy, Research Organization for Energy and Manufacture, Research Organization for Earth Sciences and Maritime, Research Organization for Life Sciences and Environment, Research Organization for Electronics and Informatics, Research Organization for Social Sciences and Humanities, Research Organization for Archaeology, Language, and Literature, Research Organization for Health, Research Organization for Nanotechnology and Material, Research Organization for Agriculture and Food, Research Organization for Governance, Economy, and Community Welfare. BRIN catalyzing research and innovation to develop an advanced scientific ecosystem in Indonesia with 2 main targets. BRIN realizes the tremendous potential of the nation's youth in the field of science and technology. Thus, various research and innovation policies are being formulated to develop and cultivate human resources with high competence and aptitude in science and technology. Creating many excellent programs is a form of BRIN's commitment to researchers, innovators, and the general population from various disciplines across the nation. The research and innovation ecosystem quality improvement in Indonesia is also realized through the collaboration of various ministries, institutions, domestic industries, and regional governments, to name a few, by forming the Regional Research and Innovation Agency, or RIDA. Moreover, cooperation with various foreign research institutions continue to be developed through active participation as member of various organizations and international forums. BRIN organizes research into a more effective and efficient process by integrating human resources, infrastructure, and budget from various research and development institutions. Oriented by a national research priority, BRIN routinely enacts comprehensive evaluations from various research and development institutions. This effort is supported through the continued management and improvement of various research and innovation facilities located across 17 regions of Indonesia, dedicated towards the development of science and technology. BRIN continues to improve our contribution to research and innovation towards national development by developing and revitalizing 4 areas of science and technology, or KST. An integrated area consisting of research laboratory facilities, testing laboratories, and industrial facilities for both startups and SMEs, tenant office rooms, co-working space, meeting rooms, gallery rooms, and other supporting facilities. manifest the future of a golden Indonesia 2045 by consolidating science and technology resources and creating a resource ecosystem and research-based economical foundation. Transforming Indonesia as a global reference in the development and application of science and technology, practices, and the good of humankind. Nature is the source of inspiration. Knowledge is the foundation, and to be put in praxis for the sake of the nation.
Good morning. Good morning, Mrs. Puji, Mr. Satrio, Professor Ross, and Dr. Tuti. Nice to meet you. Nice to meet you all of you this morning. And thanks for joining today. And we will start the webinar now. Okay. Good morning, ladies and gentlemen. May I have your attention? We would like initiate the webinar, but first, for the convenience during the webinar, please mute your communication device. Thank you. The Honorable, the Chairman of Research Organization for Agriculture and Food, the National Research and Innovation Agency, or BRIN, Mrs. Puji Lestari, PhD. Honorable, the Head of Research Center for Food Technology and Processing, BRIN, Mr. Satrio Kridowahono, PhD. The Director of Center for Applied Water Science at the University of Canberra, Australia, Professor Ross Thompson. Researcher of Research Center for Food Technology and Processing, Dr. Tuti Hartati Siregar, and all participants. Ladies and gentlemen, my name is Yuri. It's a great pleasure for me to welcome every one of you to the international webinar NgajiTekProP 18, entitled Chemical Hazards, Transfer from the Marine Environment to the Food Cycle, Thursday, September 26, 2024. Ladies and gentlemen, the exposure of contaminants into the food cycle is significant concern in public health as it directly links to the safety of humans from seafood consumption. How it affects food safety, especially from the ocean source. become the topic of this webinar. Ladies and gentlemen, please be prepared for the first agenda, the National Anthem of Indonesia Raya. Thank you. Ladies and gentlemen, we will start the webinar with warm introduction by the head of Research Center for Food Technology and Processing BRIN. Please welcome Mr. Satrio Kriwahono, PhD.
Okay, thank you, Mr. Yuri. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Excellency, Dr. Puji Lestari, Chairman of Research Organization, Agriculture and Food BRIN, honorable speakers, firstly, Professor Ross Thompson, good day, mate. It's Australian from the University of Canberra, Australia. The second one, Dr. Tuti Hartati Siregar from PRTPP BRIN. The moderator, Dr. Adestia Triwibowo. And distinguished participant and the committee of this international webinar, NgajiTekProP number 18, Chemical Hazard Transfer from the Environment to the Food Cycle. Firstly, I would like to welcome all of you and send many thanks for attending this international webinar. And I also would like to acknowledge for support from the committee, so this international webinar can be delivered very well today. And secondly, as part of my welcome speech, I would like to introduce PRTPP BRIN, or Research Center for Food Processing and Technology. I will share the presentation first, sorry. Today, PRTPP BRIN, Research Center for Food Technology and Processing, hosting international webinar NgajiTekProP number 18. PRTPP BRIN established since 2022 under the BRIN and exactly under the Research Organization for Agriculture and Food, BRIN, as one of 7 research centers in this research organization. PRTPP BRIN has a responsibility to improve, to conduct research in food, food food development, food research, food processing, and food technology, particularly because Indonesia is one of the country with highest biodiversity, particularly if we combine between the biodiversity in land and marine. So with Indonesian resources in biodiversity, we need to utilize all of Indonesian resource, particularly for food product, because we can find many kind of species for example, for carbohydrate resources, we can find many kind of species for protein, for fruit, vegetable, nuts, and herb and spices. So, as part of PRTPP BRIN responsibility, how to utilize it to give benefit for people or for economic development in Indonesia. In the last 3 years, since 2022 until 2024, we have three strategic programs. The first one is food security revitalization, particularly for stunting, to overcome stunting issues in Indonesia. And we already changed the issues in 2024 into functional food. The second one about processed food packaging and preservation, and the last one is about halal research and innovation. Why we have a research focus in halal? Because Indonesia is one of the biggest Muslim population in the world, but not only for that, but we need halal to support economic development in Indonesia, particularly in industries. PRTPP BRIN has 126 active researchers and divided into 16 research group. And we, as you know, our research group in PRTPP BRIN and our researcher, we ready to collaborate with everyone, every stakeholder, not only with campus, not only with the university, but we also ready to collaborate with industries. In the last 3 years, we already produced more than 500 international publication in journal, book, and proceeding, and produced around 100 patent, and some of them already licensed by industries. The first one, heterogeneous canned food. There are 4 industries use these technologies in Indonesia: native chicken egg flour, sagu instant noodle, anthocyanin beverage fodder, and artisan spirulina cheese. Hopefully, in further, we will have more technology can licensed by industries, and also can utilize and applied by community or people. This is our research facility inauguration by Vice President of Indonesia
Fasilitas riset pangan
in 2022.
di Pusat Riset Teknologi dan Proses Pangan, Badan Riset dan Inovasi Nasional sebagai laboratorium rujukan riset halal Indonesia, saya nyatakan diresmikan.
This research facility is not only used by PRTPP BRIN researchers, but also open for the stakeholders. Maybe if there is another, there are researchers from another institution, we are very welcome and open to make collaboration research in these facilities. To deliver or transfer technology, PRTPP BRIN has many ways. One of them by conducting webinar series since 2022, and now we already has 18 NgajiTekProP today, and we still has another 2 in this year, NgajiTekProP number 19, and hopefully next 2 months with one of speakers from Austria, and then the last one, NgajiTekProP number 20, about our implementation and collaboration research with stakeholder. For the next 5 years, PRTPP BRIN has 4 strategic program. 3 of them are almost similar with the previous one, but another is about high protein diversification, particularly to support the new government program in the next 5 years. For our discussion today in this international webinar, I wish we can get some insight, some idea, particularly in the third strategic program about halal and food quality, because this topic related to food safety, I think, food safety and some technology related of that detection and something like that. So hopefully with this international webinar, we can get more idea, inspiration, and collaboration from all of the audience and maybe from the speakers. Hopefully we can collaborate in further for that topic. And finally, on behalf of the committee, I do apologize if there is inconvenience and shortcoming in this international webinar, and hopefully every one of you can get fruitfully discussion and get new inspiration and new collaboration in further about about this topic for research and maybe for some project in further. And thank you for your attention. PRTPP BRIN, Technology for Food for Future. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Mr. Dr. Satrio, for your introduction of this webinar today. And ladies and gentlemen, the next agenda is welcome speech from the Chairman of Research Organization for Agriculture and Food BRIN, and officially open the webinar. So to Dr. Puji Lestari, time is yours.
Thank you, Mr. Yuri. Assalamualaikum warahmatullahi wabarakatuh. Can you hear me? Yeah. Good morning, everyone. Honorable Head of the Center for Food Technology and Processing, Dr. Satrio Kridowahono, distinguished speaker, Professor Ross Thompson from the University of Canberra, Australia. Nice to see you in this webinar. And Tuti Hartati Siregar, PhD, researcher from Food Technology and Processing Research Center, and chair of this webinar, Dr. Aditya, all participants, ladies and gentlemen. Representing ROA, Research Organization for Agriculture and Food, it is my great pleasure and honor to welcome all of you to this international webinar, NgajiTekProP 18th, with title or topic, Chemical Hazard Transfer from Marine Environment into the Food Cycle. First, so let's thank to Allah for his blessing that today we are gathering for this webinar. Ladies and gentlemen, as a background a bit for today's webinar, as we know that the ocean and land are crucial food sources for living organisms, including human. The sea contains a vast array of edible species accepted by almost all ethnics and beliefs. In many nations, especially archipelagic nations like Indonesia, the ocean serves a central role in addition to agriculture to ensure food security. Despite its vital roles, the ocean often serves as the final disposal of hazardous elements from either anthropogenic or geogenic activities, industrial discharges, agricultural runoff, sewage, and urban waste water released to the riverine system will end up in the ocean and contaminate the marine environment. Hazardous chemicals may dissolve in the water, sink deep in the sediment, transfer to living organisms, and finally distribute into the food cycle. So the exposure of contaminants into the food cycle is a significant concern in public health as it directly links to the safety of humans from seafood consumption. And chemicals at trace concentration in the marine environment are usually not a significant hazard, but some chemicals are essential for humans in a low concentration but become toxic if they are in the excess. However, some elements such as mercury and arsenic are not known to have living organisms, even though they are present in trace concentration. Due to their high toxicity, the concentration of both elements into surface is controlled to the level of not exceeding the limit. Therefore, the standard to set the maximum tolerable intake is based on its bioaccumulation. So how the contaminant transfer and how it affects food safety, especially from the ocean source, become the topic of this webinar. So ladies and gentlemen, with this justification, hopefully in this webinar, we could share knowledge of research on this topic of chemical contamination in the marine agriculture and how to tackle the challenges for food safety and how to broad knowledge about marine and agricultural pollution and their influence on food safety. The important thing also as mentioned by Pak Satrio, to strengthen networking and initiate collaboration and cooperation research in the future. So, with Bismillahirrahmanirrahim, I open officially to the webinar NgajiTekProP number 18. And again, I thank to Dr. Satrio Hano for organizing this webinar. Thank to the webinar organizer and all participants who made this successful webinar. Have a fruitful discussion and have a good day. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Warahmatullahi wabarakatuh. Thank you very much, Dr. Puji, for your welcome speech and officially open this today webinar. And ladies and gentlemen, and now we will have the documentation session for all the participants. So please open your camera or video, and please the host can help to take the pictures, and I will count take a picture first. Okay. So if you're ready, please open your video, your camera, and I will count, and please the co-host help take the picture. Okay, 1, 2, please keep smile. Okay, again, once more, 1, 2, and 3. Okay, the next layer maybe, co-host. Okay, 1, 2, 3, keep smile. Okay, next layer, the last layer, 1, 2, and 3. Okay, I think it's enough. Thank you very much. Okay, so ladies and gentlemen, let's move to our main agenda in the webinar today is speakers' presentations. And let me inform you that today we will have 2 speakers. So may I kindly invite our moderator today, that is Dr. Rades Triwibowo. Hello, Dr. Rades.
Halo,
How
Pak
are you
Yuri.
today?
Can you hear my voice?
Yeah, it's very clearly. Okay.
Okay, thank you.
Are you okay? In Cibinong,
Yes, all good. Yes.
Okay, nice to meet you,
Nice
Dr.
to meet you too.
Dades. And let me first let me introduce our moderator first. Dr. Dades is a food safety researcher at the Research Center for Food Technology and Processing (BRIN), and he has expertise in microbiological risk assessment, predictive modeling of pathogens, and molecular biology. And he has authored many scientific publications on the microbiological safety of fisheries products, covering areas such as bacteriology, virology, and parasitology. And he also involved in several environmental studies to ensure the optimized to health of peoples, animals, and ecosystem. And he has extensive experiment in managing microbiology and molecular biology laboratories in both private and government bodies. Okay, I think that's enough for my explanation. And now, please, Dr. Rades, time is yours.
Thank you, Pak Yuri, to introduce me and also handing over the screen to me. Good morning, everyone. Welcome to our webinar with the title Chemical Hazard, Transfer from the Environment to the Food Cycle. So my name is Raditya Tribowo, and I will be your moderator for today's session. We are delighted to have you join us on Zoom and also in YouTube streaming on BRIN Indonesian channel. So before we begin, I would like to remind everyone to keep their microphones muted during the presentation to avoid any background noise. If you have any question, please raise your hand in the Zoom during the Q&A session. And at the end of the second presentation, or type them in the chat box and we will address the question sequentially. All right, before we get started, let's do a quick check in with our speaker, Professor Thompson and Dr. Tuti. Are you both with us?
I am.
Okay.
Yes.
All good. Great. It's good to see you both again. And thank you for your time to sharing the knowledge with us. And Professor Thompson, how is the weather in Canberra today? Is it freezing?
It has
Always?
been rainy. It has not been freezing. It has just been
Okay.
rainy and miserable. So I'm a bit jealous of you guys up in Indonesia in the warmth. So
Yes, because we are tropical also, so I think it's wonderful. So just to confirm, will you both be share your screen for your presentation?
Yeah.
Yeah. Okay. So do you have access to share screen on Zoom? Just a quick check. Okay. Okay, then so great. I think so. We are all good. So just let us know if there is a problem on sharing your presentation. For your information, that the presentation and discussion will be divided into 3 sessions. The first session will be Prof. Thompson presentation, and the second will be Dr. Tuti presentation, and then the Q&A session. Now let's dive into our exciting topic for today. We are honored to have the distinguished speaker with us today. Our first speaker is Prof. Thompson. Could anyone please share the CV of Professor Thompson, if possible? Okay. Our first speaker is Professor Ross Thompson, who will be discussing the waving ecosystem function in indigenous value into agricultural landscape in Australia. Professor Thompson is a renowned expert in ecology and environmental science. He is the foundation director of the Center of Applied Water Science and the co-director of the Institute for Applied Ecology at the University of Canberra, Australia, and he co-leads the Basin Scale Environmental Flow Assessment Program, or we call it FLOW-MR, for the Australian Commonwealth Government, which is a board member of the Southern New South Wales Drought Resilient Hub and a lead counselor for the Biodiversity Council. Professor Thompson has published over 130 peer-reviewed papers and supervised more than 50 graduate students. And he is actively engaged in research on water management and the whole of landscape scale, and works closely with manager and policymaker in Australia and globally. Now, I would like to invite Professor Thompson to begin his presentation. Professor Thompson, the screen is yours. Thank you.
Thank you very much. Just share my screen with you. So although we've all been doing this intensively, every time I share a screen, I'm always a little bit nervous about whether it's going to work. So hopefully you can see my screen now. Tuti, can you nod if it's okay?
Yeah, it's good. All good.
Excellent.
Professor.
Okay.
Yeah.
Well, thank you so much for the opportunity. This is one of those wonderful opportunities to speak to a diverse group of people, and also to have involved with me Tuti Sirigar, who is one of my PhD students and a good friend. And Tuti is a fantastic researcher who worked with us here at the University of Canberra. We miss her desperately now that she's gone back to Indonesia, but it's exciting to be able to interact in these sort of forums. So selamat pagi, and I'm going to talk about something perhaps a little bit different to what you normally hear about. I do a lot of work in productive landscapes and trying to understand how we can bring the need for agricultural productivity, or sometimes coastal productivity as well, together with other demands that we're placing on the environment. So I'm going to probably be a little bit further away from the food science than many of you are used to. I'm going to talk much more about the ecosystems and the ecology. So hopefully that's interesting, as opposed to a little bit too different. So hopefully you enjoy that different perspective. I'm going to apologize for my accent before I try and attempt this, and thank Tuti for providing these words just as a way of starting. So jalan-jalan ke kebun binatang, lihat burung, cendrawasih, para hadirin harap tenang. Saya Ross Thompson akan mulai presentasi. Now, that was almost certainly the worst Indonesian you're going to hear today. Hopefully, I didn't say anything with such a bad accent that it's become, a rude word or insulting someone's mother. It was the best I could do. So, thank you very much for that. As is traditional in my part of the world, I'd also like to begin by acknowledging the traditional owners of the land on which I live, which in this case in Canberra is the Ngunnawal people, and I'd like to express my respect for their elders past, present and emerging. I'll also be talking today about some work that I have done on other Indigenous owned land, work carried out on the land of the Arrente people in Central Australia and the Kamilaroi people of Northern New South Wales in particular. And I've been gifted some words by Kamilaroi elders that I'll just speak very briefly here. So Yama Ningen Ayu Ross Thompson, Dahawan Nahale, Bradley Mogridge, Ningaya, Winingali Hana, Wayama, Wunangai, Nigyani Ganu, which means my name is Ross Thompson. It's my pleasure to be with you. I acknowledge the elder who shares their traditional knowledge through me, which is Bradley Mogridge, and thank you for the chance to speak to you today. Now, I think we're all aware of the global challenges that we work within in terms of context. And so if we look at global food security, we can see variation across the globe in some areas that are experiencing genuine tensions. If we think about energy security, similarly, we see variability across the planet, and many of the places that are experiencing food security issues are also engaging with energy security challenges. And then if we look at water security, again, we see a lot of variation, and we see some places that are experiencing water security issues already, and in some cases, those are becoming quite profound. And of course, the challenge when we talk about food security, energy security, and water security is that they come together in what we call the food, energy, water nexus. And that we can use water to generate energy, we need to use water to support our food supplies, and we also need to provide drinking water and potable water for people to live with. And so that challenge plays out through a group of different physicochemical systems. And that includes our climate systems and changes in our climate systems, which are presenting significant disruption to the food, water, energy nexus. Society and community values in terms of what is acceptable, and we see that particularly in terms of debates over what is appropriate as a way of generating energy. You know, large hydro schemes around the world have been very, very controversial. The use of nuclear power is very controversial. And so as a consequence, the food, energy, and water nexus has to address issues to do with society and community values. Local and global economics and how that plays out through that cycle. And finally, biodiversity and how we protect biodiversity. And as was mentioned in the introduction, you know, you're very fortunate to live in Indonesia, which is one of the places that has this astounding natural biodiversity. And that provides wonderful opportunities and wonderful values, and also provides some challenges as we try and feed growing populations while at the same time protecting and maintaining biodiversity. So global water security in this context, and one of the things that you'll recognise immediately is that Indonesia and Australia share challenges in that water security space. And so I'm going to focus in on Australia, which is where I live right now, and I'm going to focus in particular on the southern part of Australia. Now, Australia is the world's driest inhabited continent, and probably more so than that, it's characterised by really extreme temperature and hydrologic variability. And so it's not just that it's particularly dry, it's that there are very wet years and there are extremely dry years. I focus particularly on the Murray-Darling Basin, which is a catchment of about 1 million square kilometres, about 14% of Australia's total area. It's a very large, very long river, and it's a critical agricultural region for Australia's productivity. It's big, it's about 4 times the size of the United Kingdom, or about half of the total land area of Indonesia. So it's a very, very big catchment and incredibly significant for Australia in terms of food production and in terms of social and economic values. Just to give you some indication of that, the Murray-Darling Basin, despite being only 14% of the country's total area, produces about 98% of Australia's rice, 93% of Australia's cotton, and is also really important in terms of irrigated food crops, horticulture, and dairy production. Total population in that area, and by Indonesian population density standards, this is going to sound like a very, very low number, about 2.5 million people living across the basin. 30 billion of agricultural productivity annually, and about 40% of all Australians' farms are within this catchment. So this is a really significant area, and it's experiencing some tensions at that junction between food production and how do we manage the landscape for other values. And I've already mentioned the challenges around hypervariability and variability in the environment, and the Murray-Darling Basin is a particularly good illustration of that. So these are the flows coming down the river in gigalitres in any year. So you can see in a really wet year, so you can see there in the late 1950s, early 60s, you know, the river is a really substantive river. 40,000 gigalitres is a very large river. But in the dry years, shown there by particularly the little red symbols, you can see that it's actually a very, very small river indeed. And that variability makes food production a massive challenge, because in the very wettest years, the Murray-Darling Basin is exceptionally good at producing horticultural crops, rice and cotton. In the very driest years, it is not possible to produce those crops. And so that fluctuation has caused significant management challenges. The other important thing to note from this graph is the changes in patterns of rainfall over time. So the red dotted line that I've just animated is the median flows from 1900, when our first good records start, through to about 2000. And the important thing to note is this dramatic step change in flows. And so the area has become not only variable, but drier on average as well. And Australia is in fact characterised by periodic drought. And you're not going to see the detail. These are little pictures of Australia, and they show rainfall in red, in blue, which is higher than average rainfall, or red, which is lower than average rainfall. through the decades of the 20th century. And the most striking feature you'll notice is these large periodic droughts. Those droughts are often ended by quite extreme rainfall events. And so this fluctuation between dry and wet cycles is a character of Australia's food production system. In particular, you'll notice starting in the late 1990s, the Millennium Drought, we believe this to be the driest decade in 3,500 years in Australia. So this is a particularly extreme drought event. It was broken by extreme high rainfall, and then we had another drought that no one's named yet. So if anyone wants to come up with a brilliant name for Australia's most recent drought, email it to me. This is an incredibly intense drought. This is the most intense drought event in recorded Australian history, and that again broke with extreme rainfall. The overall trend within Australia is an interesting one. In the north and west of the country, it's increasingly being affected by monsoonal events. And so actually the rainfall in the north and west of Australia has increased over time as a consequence of climate change. But in the south and east of the country, there's an extreme drying trend. And so the Murray-Darling Basin, which I showed on the map before, sitting down in the southeast, it's really being affected profoundly by reduction in water supply. And this is happening on a system which has already had substantive amounts of water removed from it for agricultural purposes. So the red line shown here is the amount of water coming down the Murray River from 1998 through to 2008. And that red line is the actual amount of flow. We can model how much flow would have come down the river without dams and without irrigation takes. And you can see that really substantive difference. And so the river is running at about a third of what it would have done without the effects of agriculture, mainly agricultural development. So because of that, really since the start of the 2000s, there's been a program of water reform to try and bring together the agricultural community and the irrigated agricultural community together with a broader suite of land managers to try and make the use of water in the Murray-Darling Basin more sustainable. And that involved development of water trading markets in the early 2000s, a major piece of work on ecological condition in the basin, which took place in 2008. That piece of ecological work showed that the basin itself ecologically was in very poor condition and that condition was continuing to decline. The colours here are a little bit counterintuitive, but the purpley bits are in good condition and the yellow and green bits are in poor condition. And so you can see that this is a system that ecologically is really quite stressed, and that's largely as a consequence of use of water for agriculture. So if we look across the basin, 20 of the 23 valleys in the basin are considered to be in poor or very poor ecological health. So I'm going to talk about the challenges of managing this production landscape in the context of three different drivers. And there's three drivers which are now playing out into our agricultural landscapes that we haven't considered historically. Really, for the last 100 years, the focus has been on agricultural development, particularly development of irrigation infrastructure. But increasingly, there's concern about how do we bring other values into our agricultural landscapes. The first of those I'm going to talk to quickly is the issue of biodiversity and biodiversity management. Now, because Australia had relied heavily on irrigated agriculture, it had a large amount of technology really installed in the 1950s. But that infrastructure had become increasingly inefficient. And the recognition was that if we could improve the efficiency of Australia's agricultural industries, then we could take some water back from agriculture and provide it to the environment to support biodiversity. And so from 2007, there was a major program, many billions of dollars spent on modernising irrigation agriculture and irrigated agriculture. That included lining irrigation ditches, telemetry in order to record amounts of water and to manage water more efficiently. In addition, a suite of water allocations that had been allocated but which actually exceeded the amount of water present in the environment were purchased back by the government to be reallocated to the environment. This overall program was about a $13 billion program, so it's a gigantic piece of water reform. And though you can see the sort of the spread of the investment there, which was a lot of it was around investing in new infrastructure, but also purchasing back all water allocations. Now that water was then provided for the environment, and we needed to understand how much water the environment actually needed to try and redress this balance between agricultural water and environmental water. And so in 2010, there was an extensive set of investigations done, which was trying to identify what the best available science was to tell us how much water we would be able to take out of the river and use for irrigation and still protect biodiversity values. Now, the aim here was to ensure that we continued to be able to use water for agriculture, but to not have a negative effect on natural environments. Now, obviously, we didn't know everything about every part of the basin, and so what we did was look for where we had the best available science on parts of the landscape, and we created this term called umbrella environmental assets. These were places where we knew a lot about the environmental conditions in that place, and what we tried to understand is how water might change those environmental assets, and assuming that if we managed for one suite of environmental assets very well, then other parts of the landscape would also benefit. And so these are information-rich areas where we knew a lot about the environmental values, and we could work out what the water requirements were for a particular valley or region. Just to give you a sense, this is the Murray-Darling Basin, again, the outline shown there, and this is some of the sites where we carried out ecological investigations over about a 5-year period, trying to understand how much water these particular parts of the landscape needed so that we could then scale that information up to a whole of basin scale. This was done through something called the ecological elements method. There's quite a lot of modelling involved, and I don't want to spend an awful lot of time talking about modelling today because I don't want anyone going to sleep on me. But if you are interested, we've produced a paper a few years ago, which is deliberately designed to summarize this in a relatively understandable way for people who may not be ecologists or hydrologists, but are interested in that whole issue of water for the environment versus water for agriculture. So the Murray-Darling Basin Plan explained how much water that was needed, that was able to be taken out of the environment while still protecting environmental values, and that value came up as something that's 3,650 gigalitres. Now, this whole process around deciding how much water should be available for agriculture was not done in a way which it consulted with farmers or the agricultural sector very well. And so as a consequence, when this number was released, lots of farmers set it on fire. So they were angry farmers, and there was a lot of major protests throughout the basin from the agricultural sector who really hadn't expected this to be quite such a dramatic change in the amount of water available for agriculture. As a consequence, we then went back and worked extensively, particularly with agricultural communities, but also with social scientists and economists to try and see if we could understand the impacts of the sustainable diversion limit and then implement this water planning program efficiently. The Murray-Darling Basin Plan, which gives effect to that, is about trying to work out how much water we can allocate through the sustainable diversion limit to agriculture while not impacting on the environment. And in the process of moving from that sort of initial effort, the guide, through the main basin plan, that value of 3,650 gigalitres was reduced to 2,750 gigalitres. But really large amounts of water, this is about 8% of the total amount of water that was allocated to agriculture being taken back from agriculture and allocated to the environment. And so you can imagine that the tensions that would generate in farming communities. How would we use that water? Well, the recognition was that, you know, we've changed the way these rivers flow. We have removed a lot of the water, and so we have also removed some of the peak flows. So the green flow there is the way the river might flow now. The orange is how it would have flowed historically. We've got some water back for the environment, and so how do we use that water to try and recreate some of the flow events that would have occurred before we started putting big dams and irrigation offtakes in place? So you can see here we did modelling that said that, well, if we could just get the flows a little bit higher in August, then we would be able to get improved health in river red gums, which are one of the major tree species. And so we've used some environmental flows here to top up the natural flows. We also knew that the flows were not staying high long enough to support fish breeding and bird breeding events. So we allocated some of the water which was recovered for the environment to supporting longer sort of intermediate levels of flows so that we could support bird and fish breeding. Now, there were some real challenges in terms of the science that underpinned this. And one of them was just understanding the relationships between water and the landscape and how ecological systems actually responded to that water. And so we've done a lot of work looking at the way in which ecological condition changes with time when we are allocating water at various times of year. And so some systems were very simple. We have some environmental water, we've got a system which is in poor ecological condition, we provide a small amount of water and we see an ecological response. Some examples of that are many of our large-bodied fish species. The large-bodied fish will cruise along happily enough. If you give them a decent flow, particularly in spring or summer, that will provoke breeding. And so you can then generate breeding events and increase the population of these fish species. Some examples are rather more complex, however, and so if we look at something like river redgums, our major tree species, what they need is a small amount of water provided early in the season, and then larger floods provided later in the season. So in terms of how we actually manage this environmental water, it depends what we're managing for, if we want to try and get biodiversity values back into this predominantly agricultural landscape. The other challenge we had was understanding that given these systems had been degraded primarily by agricultural development over the last century, how quickly would they bounce back? And when could we expect to see responses? Now, politicians tend to be impatient people in general. And so when they've spent 16 billion on water reform, they want to know when they're going to see good returns for their $16 billion investment. And so we've done a suite of studies trying to understand how quickly various parts of the fauna can respond when you provide them with water. This happens to work on frogs, but it just tries to give us some advice that we can then pass on to politicians around saying, okay, you're really not going to see a response for 5 years, but after that you should start seeing something. And so just to encourage them to be a little more patient. We've done a bunch of work, a number of studies on a variety of different groups ranging from frogs to woodland birds through to floodplain birds. And ultimately, we've taken that information and we're now using it to guide water managers in how and when to use the water they have for the environment. So we've learned enough that we can now say that if we want to manage for floodplain bird populations, when do we need to put water into the landscape and where do we need to put it in the landscape? And in many cases, what we're trying to do is provide enough water that we can get these species to thresholds where they are going to start breeding. And one of the characteristics of these systems is that as water has been taken out for agriculture, the small and medium floods that actually provoke breeding activity in many of the fish, birds, and plants, those types of flows have no longer happened. And so we're now spending time planning use of environmental water to try and improve conditions such that we see breeding events in native species. Oops, sorry. And again, we've been trying to focus on areas which have been particularly degraded. This picture on the left you're seeing there are what should be high value river floodplain forest, predominantly river redgum, an iconic tree species. And you can see there that the removal of water so that system hasn't flooded for about a decade in that picture. And you can see the degradation in these systems that should be relatively biodiverse. And we're now getting quite a really much more sophisticated understanding. This is a very graphic summary showing what values we get back from when we can put water in different parts of the channel. So what are we targeting if we have relatively little water to use versus what are we targeting if we can get water over the banks and onto the floodplains of rivers. And if you're interested in burrowing deeper into this sort of information, the little hyperlink I've provided down there is to one of our websites, which has a lot of interactive materials for those that are interested in following through some of these ideas. That sort of visualization has also allowed us to start modeling much more effectively in terms of showing people in the farming communities in particular actually how we're managing and what we're managing for. So they can now go to a dashboard where they can see an environmental flow coming down through a river system. They can see what it's targeting, and they can see whether that target group is responding. And that's all available as an online dashboard that we share with particularly colleagues in farming communities. For the general public, we have been really trying to connect with the general public in terms of understanding why it is that we might need to get water back for the environment, even in agricultural dominated communities. And again, if you want to go and see some of that sort of information, which is more targeted at the general public, please go to the website and have a look at it. So biodiversity in an agricultural river. One thing we've really learned is that it's pretty possible to start to manage these systems for biodiversity, but there are trade-offs. And one of the challenges that we have had is trying to convince communities that there is a need to trade off some agricultural productivity in order to try and protect biodiversity. There's a tendency to say that there's only going to be winners out of changes in water management, coastal management, or land management. That's usually not true. There are normally trade-offs, and it's important to be able to communicate those. Okay, now I want to talk a little bit about agricultural landscapes and the role of managing carbon. And again, this is in the Murray-Darling Basin, but I'm going to focus in on the southern part of the Murray-Darling Basin in the state of Victoria. So Melbourne, the largest city in Victoria, and we've been really fortunate over the last 30 odd years to have a really extensive program of measuring aquatic biodiversity, mainly aquatic insects, across the state of Victoria. Again, if you're interested in the details behind that, there's a global change biology paper there that you can look up. These landscapes have been quite severely modified, so they've been cleared. They'll naturally be woodland systems, and particularly around the streams, we've lost native vegetation along the edges of the streams. And the opportunity was identified that maybe we could restore some of the forests in this landscape, and that would allow farmers to generate carbon credits. And so they would be diversifying their farming from pasture land to pasture land plus carbon farming. And so that's the sort of diversification and change we're interested in. And so turning streams from something like what you see on the left there to what we see on the right there, relatively small bands of planted trees, but in quite a productive part of the landscape. So they should suck up carbon quite effectively. In order to investigate this, this is a patch of central Victoria. We had paired sites where we had either areas that had been cleared or areas that had been replanted over the last couple of decades. I will give you some sort of ideas of what they look like. Here we have a stream. We're upstream. Down the bottom of the picture there, you can see there's no real trees. When you go further downstream, you can see that there's replanted trees. You can see them in the landscape. So we've got sites which really have almost no trees at all. We have sites that have been planted relatively recently. These are 6 years old. By Indonesian tree growth standards, this is pretty embarrassing. A 6-year-old tree in Indonesia is looking pretty happy by now. This is a very dry landscape. It only gets around 200 to 300 millimeters of rain a year. And in some years, it gets down less than 100. So it's extremely dry. But there you can see if you plant trees, they will very slowly grow. After 8 years, we start to see some of the larger trees coming in. These are then a 12-year-old planting. And if we get up to 19 or 20 years, then we start to see sort of canopy closure over the stream. And what we were able to do was to go out into these different ages of plantings and measure where the carbon was going within that ecosystem. And so a series of calculations which took place, which measured carbon on the ground, carbon coming in from the trees. We measured how much the trees were growing using dendrobands so that we could measure actual carbon accrual. And we measured how the carbon was moving in and out of the water column using metabolism techniques. What that let us do was to take a piece of the landscape and then model different options. So we could say, okay, here at the top figure, this is how much carbon would have been in the landscape in pre-clearance time, so before this area was cleared. All right, we've now cleared most of the lower parts of the landscape. There's still forested catchments left in the upper parts of the landscape. We now know how much carbon we are removing in that scenario, and we can then explore scenarios where we model what happens if we plant particular parts of the landscape. That allows farmers to make some decisions about whether it is more effective for them to retire particular areas that they're grazing and plant trees on those areas, or whether they're better to continue grazing activities. And many farmers have implemented these models now in terms of trying to diversify their farms from just relying on grazed agriculture through to grazed agriculture and carbon. So take-home messages really from that is there certainly are areas in the landscape where carbon farming can be used without compromising agricultural production. There are some challenges, however, and it's that these trees, as they grow for the first 20-odd years of their life, actually use quite a lot of water, and this is a very water-poor landscape. So farmers have to trade off the advantages of carbon farming with some of the disadvantages in terms of demands for water. And again, these are now being built into relatively complex landscape scale models where we can say what would happen if the carbon price globally doubled and more farmers took up carbon farming. What would the effects of that be on flow in rivers? And how do we effectively manage that transition from predominantly grazed agriculture through to grazed agriculture mixed with carbon farming? The last case study I want to talk about is something which might be really quite different to you, but I suspect it's something which is really relevant in all food production systems around the world, where we have indigenous groups who are present on the landscape and still actively managing the landscape. Australia is a very diverse place. I think in the introduction it said there's about 700 Indonesian languages. There's also about 700 Indigenous Australian languages across the country, and those language groups are broadly shown here in these multicolored maps, so you can see all the different groups. It's the oldest continuous culture on Earth, so we have 65,000 years of evidence of some cultural practices going on. But this has been severely affected by colonization. So of the 700 identifiable language groups, we think about 400 have gone extinct, and those languages are now lost. At European arrival, which is around the late 1770s in Australia, about 1 million Indigenous people were present. That number got down to probably in the sort of tens of thousands. But has now somewhat rebounded, so about 3 to 4% of the contemporary Australian population are Aboriginal or Indigenous Australians. It's been a really difficult history for Aboriginal Australians. Until 1967, Aboriginal people were not classified as human under the Australian legislation, so they could not own land, for example, and they certainly could not own water allocations. As a consequence, they're alienated from land and water, and there's been an ongoing process of reconciliation around that. Now, about 26% of the Australian land area is managed by Aboriginal people, but that's predominantly in the very dry, very arid parts of Australia. And in terms of actually managing water in the landscape, it's less than 0.01% of water allocations in Australia are managed by Indigenous groups. Now, this has been identified as a really significant challenge moving forward and actually a real source of political disruption. And so there's an appetite for trying to bring Aboriginal people back into water management. And the real question is, how do you do that? Because it's not actually a very easy thing to address. And so we've worked in the last 5 years now to try and develop processes which help us engage with Indigenous people in order to move to a world where Indigenous people are equal partners in managing water. And so against this really complex landscape, we've been working in one specific part of Australia, which is Kamilaroi country. This is in northern New South Wales, for those of you that know Australia well. And this is an area which is in the Gwydir catchment. And the work we've been doing is going out on country, meeting with elders and other Indigenous people and saying to them, how would you like to be involved in moving into water management space? How best can we communicate with you? What are the values that you're most interested in protecting and managing for? And how can we start to build those into our water planning processes? Now, I'm not going to spend time going through this. This is the process that we've developed, and this is particularly work done by my colleague and PhD student, Bradley Mogridge, which sort of lays out a flow chart for how we might start to engage with Indigenous people in a way which is really structured and which protects Indigenous values, but also allows those Indigenous values to be brought into water management processes in the way that they're currently done. And we've particularly been focusing on going out into Indigenous communities and identifying what values they're really interested in managing for. Now, in some cases, those values are the same as environmental values. For example, many Indigenous communities are interested in developing and protecting fish populations because they're very important harvest items for many communities. And they're also a target of our environmental water as it's currently managed. But there are also some cultural values, some plant species, some animal species that haven't really been managed for using environmental water, but which are culturally very significant. Again, if you're interested in the detail of this, this is work which has been published by Brad and myself, and which is intended to really provide perhaps some useful insights for anyone who's engaging with Indigenous groups around water or any type of resource management. So in talking to Indigenous people, there's a number of themes that emerge from the conversations that we had. One of them is that the way that we tend to manage things as non-Indigenous people in Australia is we tend to like breaking things up into pieces. We like studying water separate from land, geology separate from the ecology, botany separate from the entomology. Agricultural science doesn't always work that well with ecological sciences. And Indigenous people were really quite confused by that. I think they very much think about systems at a whole of system scale and how you manage landscape as a set of connected pieces. And so there was an emphasis when we talked to Indigenous elders that we needed to think about connecting things together. And they found it, they weren't interested in engaging with a process where we were breaking the landscape up into pieces. So there's a real strong belief in connectedness. That the belief moves across into the spiritual world, and that Indigenous people believe that there is no difference between the spiritual and the biophysical world. And so in terms of engaging with Indigenous people, there was a need to engage with their spiritual views as well. There was a reluctance to engage with us if we didn't recognize the interconnectedness of those systems. And Indigenous colleagues and friends and landowners and stakeholders really responded positively when we recognized that their values were important, as opposed to emphasizing the values just of managing water by itself. We've tried to find ways to work with Indigenous people in terms of visualization of some of those different interactions between the spiritual and the physical world. This is an example of some work that we did looking at the way in which different parts of the landscape interact with one another in the Indigenous worldview. The second thing that we've really been working with is working with Indigenous people on the longer knowledge they have, mostly recorded through oral histories. It's particularly important as a water manager because they can actually tell you over tens of thousands of years when have particular parts of the landscape had water in them or not had water in them, and help us understand which are the most important refugia at that whole of landscape scale. So there's millennial knowledge of where water persists in the landscape. And you can imagine why, if you live in a very dry landscape and you're in a very dry year, there should be really good oral records of, in a dry year, where do we go where we can be sure that there will be water present? And so there's excellent narrative histories of extreme drought events and where water can be found in those driest of times. And those are mapped through something called storylines. This is a physical drawing of a storyline. Someone would sing to you or tell you in a poem form how to navigate through the landscape to find particular places, and the circles in this diagram indicate places where there's water, and then the shading within those circles indicates where that water is present in dry, moderate, or particularly wet years. The other factors that came out of our conversations with Indigenous elders were the importance of particular species as indicators of when seasons are passing. And in Kamilaroi country, there are 14 seasons, and when those seasons occur, guides Indigenous people on when they should harvest particular food products in the landscape. Now, most of the seasonal markers are floodplain trees, and those trees are no longer flowering or fruiting at the same time as they used to because water has been removed for agricultural purposes. And so they were really interested in saying, how can we put water back into the environment that allows some of these trees to flower or fruit at the time of year they have historically? And so the loss of those cues was seen as being a really significant loss to the landscape. And the risk that many of the elders identified to us is that there's now been almost 100 years since we've had natural flow regimes in this landscape, and that if we don't do something about trying to restore some of those natural flows, that their people will forget how to harvest particular food types or how to carry out particular indigenous activities. We've also started a program of drawing out these figures. This is very complex and I'm not going to spend time going through it, but this is a seasonal calendar. It records the different seasons on country in the Fitzroy River over on the western coast of Australia, and talks about what species are expected to be flowering or producing or suitable for harvest at what times of year. The final point that came out of our conversations was the fact that for many Indigenous people, actually participating in discussions around management of resources was really difficult because they didn't have the scientific training, or they didn't necessarily understand the way in which the governance was being carried out. And there's some good examples of this. I'm sorry, that's the wrong arrow. Some good examples of this where basically you can see incredibly complex policy processes that most Indigenous people didn't feel comfortable about engaging with. And so a lack of water literacy, and this will be the same if we were talking about coastal harvesting of fisheries, the lack of an understanding of the science makes it a real barrier for Indigenous people to engage. And so there was a real request from Indigenous elders to try and generate some sort of information that might be appropriate to share with people who don't necessarily have scientific training. This is an example. This is a very simple diagram. I lied, it's not a simple diagram at all. This is a diagram where it shows the different forms and paperwork and different organisations within government that you would need to engage with if you were an Indigenous Australian person and you wanted to be involved in water planning and management for cultural outcomes. And my colleague, Brad Mogridge, who drew this up, describes it as a horrendogram. So it's sort of horrendous plus diagram combined together, really, really difficult to follow. And you can see that if you perhaps weren't scientifically trained, weren't familiar with these sort of processes, that to actually engage with this would be really difficult. So those things together tell us that actually, if we want to engage Indigenous people, then we need to think at system scales. We need to think about longer amounts of time, to think about how to restore parts of the landscape they care about, and we need to provide information that allows them to engage in an effective way. Now, none of those are particularly surprising outcomes, and in fact, many of our non-Indigenous colleagues, particular people from farming and fishing communities, have said the same, that these are the sort of things you need to start thinking about. I do think engagement of Indigenous perspectives is a really key element, and it's been a sort of a theme which is emerging around the world now, that we really do need to think about how do we bring Indigenous people into our ways of managing our food systems. Because if we don't do that, then the potential for disruption down the line is quite significant. So Indigenous values, really the take-home message is the importance of incorporating Indigenous values. And if we want to have resilient long-term management of policy, management of natural resources, then we need to have Indigenous engagement. So the take-home points I've made today, and I hope in terms of a food science perspective, we are thinking about the fact that our food science systems are all reliant on these complex agricultural systems that are increasingly under pressure to recognise social values, economic values, and ecological values, all in large complex systems. And the decisions that are made in those systems are not purely scientific decisions. They're about what society is willing to trade off. And so society may well be willing to trade off some biodiversity values in return for increasing fisheries. But is that something that, as a whole of country, does Australia or Indonesia want to make that decision? And how do we weigh up the importance of natural heritage, so biodiversity values that are under real pressure, versus economic returns that are really important to our nation's well-being? And I think as scientists, the challenge we have is how do we explain those decisions to people? So whether you're explaining those traditions to communities that are involved in fishing practices, or whether you're explaining them to farming groups that are having to give up some of their agricultural heritage and some of their agricultural productivity in return for biodiversity outcomes. The challenge for us as scientists is to actually be part of those conversations and to communicate in a way which is understandable and which really helps inform better management of all of agricultural and food systems. That's the final comment from me, and I'm happy to pass over to Tuti. Do we going to have questions now, Tuti, or do we going to have questions at the end of both the presentations?
Thank you,
I think it will be at the
Professor.
end. Yeah.
Yeah, it will be at the end of the presentation of Ibu Tuti, Dr. Tuti.
Excellent. Okay.
Okay, so you can wait for a few minutes for the Q&A session, Professor Thompson. Thank you, Professor Thompson, for that insightful and comprehensive and complex presentation. Based on your presentation, we may conclude that good quality water is essential for ecosystem, isn't it? And it's very expensive to manage in Australia, and including for farming, and perhaps similar condition in Indonesia as well. So I think there are a lot of some approach we can adopt to tackle some similar issue that might also happen in Indonesia regarding the land, water, and environment management, especially in indigenous land for agricultural activities, because here in Indonesia we also have indigenous land for forestry or agricultural activities. And anyway, we will open the Q&A session later after the presentation from Dr. Tuti. Meanwhile, we participant, everyone might type your question in the chat box, and I will read them out later for Professor Thompson to answer. But I remind you that please state your name, your institution, and to whom the question addresses. Now we come to the second presentation. Our second speaker is Dr. Tuti Hartati Siregar, who will be presenting about arsenic in human food. How much do you consume daily? And Dr. Tuti is an environmental chemist with a strong background in analytical chemistry. And before she joining BRIN in 2020 as food safety researcher at Research Center for Food and Food Technology and Processing, Dr. Tuti led the analytical chemistry laboratory at the Ministry of Marine Affairs and Fisheries in Indonesia. And during her PhD candidate, she also contributed to the management of the analytical chemistry laboratory at the University of Canberra, and was involved in the teaching first-year student about chemistry subject. Professor Thompson supervised Dr. Tuti during her PhD, and she was actively involved in adapting research to policy through focus group discussion and produce recommendation for local government and central government within the Ministry of Marine Affairs and Fisheries and the local government as well. And her policy brief was adopted to address Indonesian seafood quality and safety standard at the time. Dr. Tuti has published many scientific papers, 2 patent and 5 policy briefs. And now I would like to invite Dr. Tuti Hartati Siregar to share her presentation. Dr. Tuti, I will give you about 30-minute time. It's yours, Dr. Tuti.
Thank you, Dr. Radistia. Can you hear me?
Yes, clear.
Can everyone hear me? Great.
Yeah.
Yeah. I will start to share my presentation. Can you see that?
Yes.
Yeah, great. Okay. Rose,
Can
if
you make it
you
full screen?
Yeah.
Dr. Tuti?
Yes, yes. Yeah,
Okay,
I will
thank
do.
you.
So, yeah, it's already on full screen. So, for Rose, if you want to take a break, please do. I'm pretty sure you already know my presentation because I did it several times at the UC. And because Ross started his presentation in a poem in Indonesia, I would like also to make a pantun that I prepared this morning. I'm sure this is my the only talent that I got, that I am sure that I am better than Ross, because everything, he's my supervisor and everything I know is better than me. But for this, I know that for Indonesian speaking, I know that I'm far more better than him. So I will start with poem, with the pantun. Ke supermarket beli minyak, lihat Vegemite berbau terasi. Bapak Ibu harap menyimak, giliran saya Tuti yang akan presentasi. Okay, this is my presentation about arsenic in human food. So at the end of my presentation, we will try to roughly calculate how much we consume arsenic daily in our food. So to start with my presentation, I would like to share the knowledge about arsenic, so we are on the same page about arsenic, because I can see in most of Indonesian paper, arsenic is it's put into the heavy metals. So most of authors write arsenic as part of heavy metals. In fact, if we go back to periodic tables, this is the table. I'm pretty sure all of you understand the table, at least during your high school. So it's a bit different now from when I got this periodic table when I was in high school. So now it's from group 1 to group 18, arsenic in group 15, or we call it nitrogen group. Arsenic is in the 3rd row of the periodic table. So if you can see here, if we talk about heavy metals actually here, most of them are in transition group, but some of them are here. So if you call like lead here, or mercury, or silver, or cadmium, or copper, they are all, we call it heavy metals, hazardous heavy metals. So we call it heavy metals because they have a high atomic number. So arsenic and everything here in the gray, they are not metal. They are not non-metal. We call it metalloid. That's why they share some of the properties similar to metal, but some of properties similar to non-metal. That's why most of elements here in this group, most people, before they know the hazards of the element, they use it in their everyday life. And arsenic as a mineral, so arsenic actually God already keep arsenic deep in the earth, so no one can touch it because this is toxic. But because of the something happened in the nature, or because of the greediness of the human being, they like to mining or doing something and utilize arsenic. That's why we can see arsenic in our environment now. So the abundance of arsenic is actually very low. So it's in the rank of 54th in the earth crust, and 28 in the seawater, and 30 at the 31st number in the human body. So it's very low. And the concentration in the crust is also very low. It's about 1 or 2 ppm. That's why it's called as a scarce element. Arsenic is never found alone as a single element in nature. Usually there is impurities when someone wants to mine arsenic with bismuth or antimony, and iron, nickel, silver, sulfur, and selenium. And arsenic also have mass. It's about 75, but there is no isotope for arsenic. So if you see isotope has the same proton number, but there is a different neutron number which make the mass different. But for arsenic, we can only have in around 75 mass. So this is important when we do the analysis doing mass spectroscopy. So we will use this mass information. But although arsenic have only one isotope, 100% at the 75, the arsenic have a different oxidation number, the variation of the oxidation number from -3 to +5. But there are two oxidation number that commonly known as +3 and +5. +3 we call it arsenide, and +5 we call it arsenate. This different oxidation number will make arsenic have a range of chemical bonding. That's why in later on in the metabolism or in the environment, or in the not in the environment, in the organism, we can see that arsenic have a different molecule, not only single molecule. The sample of arsenic mineral, the most abundant is arsenopyrite. It's the bonding between iron, arsenic, and sulfur. And then the other two look like mice. One looks like ruby, also called ruby sulfur, but actually it's very brittle. So, because of the brittleness, it's often made like a powder for the application in the human life. And then we also have that's bright yellow. We call it orpiment. Now, because of the properties of the arsenic and the bright color of the arsenic, in ancient era, arsenic usually used as a dye in a painting or in the material like fabric or in the wallpaper. During the industrialization era, arsenic also used in pharmacy, also used as traditional medicine, also used in a cosmetic. And although in most of country arsenic is already banned, the use of arsenic is already banned or reduced, but in several countries, including Indonesia, arsenic is still used in agriculture. So it's used for mixing pesticide, and also with the poultry feed, and also for the semiconductor in the electronic industry. And how arsenic arrive in our food? It's as I said before, it's from nature activity. Say it's eruption from the volcano, but most of arsenic coming to our food is coming from our activity, human activity, or we call it anthropogenic activity, such as mining or industries like alloy or batteries. And also what we have in urban activity, cosmetic, the use of cosmetic or in fabric, everything. And then also in agriculture. So if Ross said that there is a limited number of water in Australia, in Indonesia we have plenty water, but unfortunately most of our water already contaminated with, including arsenic contaminating in our river, and then all flows to the ocean, and then make the ocean also acquire arsenic or take arsenic from the land, and also from the background that coming from the nature of the ocean. And then to continue, I also would like to tell you about the arsenic species that based on the oxidation number of arsenic. That's why arsenic have different molecule where it can bonding to such as molecule, organic molecule or inorganic molecules. So we call it the different molecules of arsenic. We call it arsenic species. So it's different to the species of bacteria or algae or other organism. The arsenic species is the species of arsenic that arsenic bonding to different molecules. The hazardous arsenic here, the one here at the top, we call it inorganic arsenic. Arsenic is not attached to carbon, any carbon, so it's only attached to the inorganic molecules. This is hazardous, and this is carcinogenic. So that's why if you're talking about arsenic, carcinogenic arsenic or toxic arsenic, it's coming from these two species. And then the one here at the bottom, we call it organic arsenic because arsenic attached or bonding to a carbon, either as a simple carbon, methyl or dimethyl, or there is other functional groups such as ethanol or acetic acid, and check that. Yeah. And the other part of arsenic that commonly found algae and other marine plant, yeah, is what we call arsenoriboside. So when arsenic attached to when arsenic attached to riboside or ribose, this is ribose, what we call it, or we sometimes call it as sugar. It's a type of sugar. So this is another part of arsenic, and it's also organic arsenic that we still don't know if it's a hazard or not, but to prepare or to make it to make people aware, we call it that's a potential hazard. Mm-hmm. And then the last one, arsenobutane, that's we found almost 90% in marine fish. We call it arsenobutane because it's similar to butane, what we have in our body. And this arsenic is non-hazard. So I tell you about the arsenic species because this is important to say that later on about the arsenic standard, whether whether we want to low in one particular food, or whether we want to make it higher in other type of food. And this part of my research during my PhD. So we make a small what you call food cycle, ocean food cycle. And then also from other literature, we found it's this cycle of arsenic in the ocean. So arsenic in water and sediment is very low at the background, or say that's a very clean area, very clean ocean. It has arsenic. It's around at around 1 to 2 ppm, very low. And then that's coming from the background, coming from the natural, or we call it geogenic activity. And then in the sediment, make it higher, up to 220 ppm. And then this type of arsenic is in the inorganic arsenic, as arsenic 3 or arsenic 5, when marine primary producer or algae, for example, taking up arsenic from the environment, from the water column or from sediment, and then metabolize the arsenic inside the metabolic system. It's become arsenic ribose or arsenic phosphorylated. And the total, we call total arsenic, we're not saying the species, the total arsenic in algae is vary from 1 to 131. That's what we have analyzed. And then, of course, herbivores say that the tritivores, omnivores in the ocean eat algae. And then they will also metabolize arsenic from the ribose form into arsenobutane and arsenic lipid. The number of the arsenic riboside and arsenobutane in the herbivore is about equal. But when we go to the carnivore, the form of arsenic mostly we found in arsenobutane, or some of them will make it because the complexity of the metabolic in the fish, arsenic will be changed or transformed into the lipid form. And then also when they excrete, when the organism excrete arsenic, it will become going back to the environment, but mostly back into the form of inorganic arsenic. And before we go to the food contamination, arsenic contamination food, I would like to share with you about the arsenic standard in several countries. It says here, most of the country put arsenic standard as an inorganic arsenic because that's the hazardous molecule species of arsenic, not as a total. But except in Singapore or some in Australia, still put rice as a total arsenic because especially for infant. Of course, we have to put total arsenic because arsenic is in infant or in like in formula, we have to reduce the contamination as lower as possible. And here in Indonesia, this is interesting because based on the regulation in Indonesia, last night I searched regulation in Indonesia, and the latest that I can find is in 2019, still the same. When we have a meeting, like in 2020 with Badan POM, arsenic in fish and mollusca from marine, they said it as a total arsenic in the concentration is very low. 0.25. I think this is, this is a very, we can't really accept it because later on we can see that arsenic, if we see from the back slide, arsenic in fish is very high, in very high concentration, up to 200. But most of the arsenic species in arsenobutane. But if Indonesian put the standard of arsenic in total, like 0.25, most of fish from Indonesia should be rejected. So we shouldn't eat fish, but it's not going to be, especially when now with the new government say that there is a milk fish that we're gonna use from fish. So of course this standard is not acceptable to me. And this is the whole Indonesian standard for arsenic. So if we can see the rice is the most important part because Indonesia people in Indonesia eat rice almost every day, almost every time when eating. So, but we don't have here the arsenic standard for rice. Instead, we have very low for fish and organism from marine. Now we continue to the arsenic in human food. So we have from human, for human food, we have from farming, say that we have cattle, or from the ocean, we have fish, we have algae, and from agriculture activity, we say that we eat rice every day to make us eat almost this type of food every day. if we compare between dietary, if you have it, only the 3% of inorganic arsenic or hazardous arsenic coming from fish and shellfish, but the most share coming from rice and cereals, and also sweets. We say that what is chocolate drink or candies. So we have it here, the most of the share of inorganic arsenic. So, but unfortunately, this standard that we don't have here right now in Indonesia, especially for rice. This is I want to calculate with you later on at the end of my presentation how much we uptake arsenic daily. So this is to make a brief overview for you. This is what arsenic in water, 100% is in the form of inorganic arsenic, and arsenic in water can be up to 300 microgram per liter. That is very high concentration, especially when the area is contaminated by arsenic. But I think we don't have it in Indonesia, but for people like in Bangladesh, China, Mexico, they have like high contamination of arsenic in their groundwater. So, especially if we drink from groundwater, so we have to take care about the arsenic concentration. And then we also have from rice, also the concentration not very high, maximum only 1.8, but most of it in organic arsenic. And then from poultry and egg, very low, but most of it in organic arsenic. Seafood share like high concentration of arsenic, up to 200, but almost 90% in arsenobutane. So it's as we said before, arsenobutane is not a hazard, it's not toxic arsenic. And then seaweed also share high concentration of arsenic, but most of in the form of arsenic riboside, or we call it arsenic sugar. We also, most of people in the city like to eat mushroom for their food, because that's called as a healthy food. Also contain arsenic similar to rice. The concentration is similar to rice, but mostly in organic form. So also the other like fruit, dairy, and cereal also contain arsenic. So this is the example of how we calculate daily intake of arsenic from our daily consumption. Say that if we have this morning, if we have breakfast cereal, this example, and lunch with this type of food, rice, there is chicken and tofu and tempeh and vegetable, and some of us maybe diet, so they skip dinner and only have coffee for their dinner. So the cereal that we consume daily contain milk in it. So we have it with the consumption rate. This is the consumption rate from the Ministry of Health. They have data about this consumption rate. So this is the average that I put it here, and then the estimate daily intake from cereal is about 1.74. Then from milk, because cereal have, in the morning we have cereal, and then we put milk into it, and then at dinner, during the dinner, we have also coffee, and then with coffee with milk in it. So we estimate like we have consumption of 200 meal or gram of milk for a day. That's why it's time for this meal. And then chicken, we have here a chicken, one portion of chicken, and then we have vegetables, rice, and from tempeh or tofu, it's like a grain. I took the inorganic arsenic calculation from grain. And the estimate daily intake of arsenic via consumption of this type of food is around 1.5 to 7 or 1.5 for a day, for today. If I consume this type of food today. So the maximum tolerable daily intake is only 2.1. How about if I have breakfast, say rice, and then lunch also rice, and dinner also rice? Of course, will be times 3. The concentration of without the cereal. So from rice, we will double it with 3, and vegetable as well. So of course, if we have rice at during breakfast, lunch, and dinner, we will exceed the maximum tolerable intake of arsenic for this day. So this is to calculate. That's why we need to make a variation in our food. So this to conclude. First, arsenic is categorized as a rare element in the earth crust, so it's scarce. Number 2, arsenic is transferred from the environment to the food cycle. And then arsenic is biosynthesized in the organism and forms various new organic arsenic molecules. This type of molecules, there is the hazard, there is a non-hazard, and there is a potentially hazard. And then also there is importance of arsenic speciation, because we can't say that this is dangerous or this is not dangerous only from total analysis of arsenic. We need speciation here, especially when we put it into standard, national standard of Indonesia. We need speciation data. We can like rely on single total arsenic analysis. And also for the take-home message, we need to vary our diet to reduce arsenic intake. So we're not doing the same food. We not consume the same food from morning into the afternoon and day after day. And this is the end of my presentation. Thank you.
Thank you, Dr. Tuti, for that informative presentation. I think it's related to the our daily life regarding to the food that contaminated with the arsenic, either from fish or from rice or from cattle, etc. So we now proceed with the Q&A session for both presentation. So for the participant, could you please raise your hand and open the camera to in Zoom, or maybe you can also directly ask the question, or maybe you can also type your question in the chat box and I will read them for Professor Thompson and Dr. Tuti to answer. And I have some question from the chat box. Maybe I will read one by one. Let me see. There is a question from Dr. Widodo Suito. He asking about the question addressed to Ibu Tuti as well to Professor Thompson. For Dr. Tuti, how food can be contaminated with arsenic, and how is the arsenic toxicity in human? Is that pathogenicity, pathogenesis toxin, or what do you call it? Or acute symptom? It is quite small. So that is the first question from Dr. Widodo. Could you respond to the question, Dr. Tuti, and then later I will ask about question from Dr. Widodo to Professor Ross, Professor Thompson.
Yeah. Okay. For the first question, from Dr. Widodo, yes, the arsenic, it's like the toxicity of arsenic is it's at some point acute if we like consume arsenic directly. So either if you want to kill, or if you want to kill yourself, or if you want to kill somebody, no one eat arsenic directly. So if it like contaminated from the nature or contaminated from the food cycle, we call it like long term of toxication. So it's not like an acute one. So that's why we have like estimated daily intake, and then and then there is a maximum of standard how much arsenic that we can consume from our food, from the range of food daily. So that's to calculate. So for example, we have 80 years of life. For the 80 years, we not like have the symptom from arsenic toxication. So that's to answer number one. And then number two, with the arsenic in rice, yes, there is a contamination in our agriculture, especially if we still use pesticide from element that is already banned in some countries, but we still use it in Indonesia. Of course, there is a contamination from our environment of the rice field. And then in Indonesia, as I said, we still rely on total arsenic. There is I think from Badan POM, they already try to go to the inorganic arsenic. They already try to do the speciation, but most of the laboratory through all over Indonesia, the speciation is still rare. So we still even at our laboratory at BRIN, we have the facility, we have the instrument, but we don't have the say that only column for the speciation. We couldn't buy it from the small research funding that we got now. So that's type of the problem that we face in Indonesia to shift our arsenic analysis from total into speciation. I think I offer to you, Ross.
Okay, thank you. Is that address all the question from Dr. Widodo? Is that address your question?
Yes.
Okay,
Thank you,
thank you.
Tuti.
Thank you. So there's another question from Dr. Widodo related with the use of species Posilopora, Damicornis, that can be used to decrease the microplastic. Is that the decrease or degrade the microplastic accumulation in the water? Do you have any comment about that, Professor Thompson?
Yeah, it's actually a really interesting question, because I think microplastics as a contaminant are going to become of more and more concern, in particular because some of the microcontaminants bind to things like heavy metals. And so the actual interaction between that. Posilopora is really interesting. If we can basically deliberately grow Bacillopora and then extract the biomass. It's a really good way of getting microplastics out of heavily contaminated systems. And so farming of Bacillopora as basically a biofilter is a really interesting idea. I'm aware of a few trials that have done that. I don't know whether it's been done in Indonesia yet. Clearly, that's something you would do while we try and manage the issue of microplastic contamination and reduce the sources. But yeah, in heavily contaminated systems and coastal waters, I think it's a really has a lot of promise. It's pretty tough coral too, which is good.
Okay, thank you. I think that's clear answer from Professor Thompson regarding the use of species Bacillopora for degradation of microplastic. Now, I ask someone, is there anyone raising the hand? Okay, I think.
Yes,
Oh, okay. Time is yours, Dr. Dwi now.
Well, thank you very much, Parades, and also Prof. Ross. Nice to see you again here, and good to the also. Well, I have a question, Prof. Ross. You mentioned in the conclusion that it is important to make a balance between a socio-economic aspect and the ecological aspect. And my question is, somehow in practice, it's not that easy, because we are now living in the very competitive time or era. So that's in some cases, so even in most cases, that the economic return or the profit oriented is more important than the ecological reason or the ecological risk. Of course, in Indonesia, in the very diverse socio background of the community, so perhaps a little bit different to the Australian. So, what is your opinion or your suggestion concerning this matter in the practical application of your suggestion to make the balance? And the second is related to the microplastic and the plastic issue. Do you think the plastic debris and microplastic that's now also moved to the nanoplastic still become the important issue on the water pollution in the recent decades? Because if I follow the progress, yes, but there is not so much significance what updates. For example, concerning the effort, like in Indonesia, we have many activities in order to protect the use of reduce the plastic debris, but also to promote the use of the alternative product. But there is no regulation, single regulation that's clearly put the limit or the maximum concentration limit either in the environment or in the food product, for example. So if there is no regulation like arsenic, arsenic is clear. There is a limit in the rice product, in the seafood product, in the seaweed product, but there is no regulation the limit of the plastic for the meantime. So what is your opinion about this? Is it still like this, or in the near future we will have like regulation which is more applicable to really reduce or protect the environment from the marine debris or plastic debris, or there is any new issues emerging contaminant in the near future, Prof. Ross? Thank you very much.
Thank you for the question. I'll answer the second question first. I mean, the microplastics issue, we also don't have food limits yet, and nor do we understand enough about long-term exposure of consuming microplastics indirectly. So we know that microplastics are occurring in the flesh of fish that people have been eating. We know in some cases that those microplastics are bound to hormone products, and so that's obviously a risk because we're picking up hormone products. But we don't have good standards. I'm actually thinking microplastics, I suspect many of the ecological implications are as bad for marine systems as anything else. And so we know from microplastic contamination in coastal fisheries that it can block the filter feeding organisms, basically fills their filter feeding mechanisms up with plastic instead of food, and then reduces productivity of shellfish beds. So I suspect that some of the ecological impacts on food systems are as severe as the toxicological ones. But it's certainly an interesting area to get into. We've just really started moving into it in my group now. The social, economic versus ecological perspectives is something that I think worries a lot of us, and that we all live in a world where economics is really the dominant paradigm. We don't think about sustainability. Even when we talk about sustainable development, the word development is always much more emphasized than the word sustainable. I think there are a couple of answers to that question. One, I think as scientists who aren't economists, we need to talk to social scientists and economists a lot more. We need to actually work with our colleagues within the university systems who work in those areas and try and come up with solutions that are genuinely triple bottom line, so they genuinely do take into account ecological cost. I think secondly that we need to be better at communicating risk to the general public. At the end of the day, politicians decide a certain number of things, but if the general public get upset about stuff, politicians change their view. And so I think we need to be talking to the general public in a way they understand about risks associated with these things. And the conversations go, do you want your grandkids to live in a world that looks pretty terrible? Because everyone, when you start talking about children and grandchildren, then all of a sudden we start to think about sustainability. So I actually, I make an effort myself, and I think we all do need to make an effort to communicate more effectively with other disciplines so that we can start to build solutions with them, and to communicate with the general public so they can be informed about the choices that are being made. Because I think too many of the choices are being made on behalf of people, assuming that they don't worry about sustainability or that they don't worry about the environment. Actually, they worry about it more than people think, but politicians aren't hearing those voices often enough.
Okay, thank you very much.
Okay, thank you, Professor Thompson, for your responding the question. So now there is question actually from two person, Dr. Christine Tia and also the Dr. Nurfianto. It's related with the arsenic in the food. The first question is, what species of arsenic are generally found in the seaweed, and what is their toxicity if consumed by human? And the second is about have there been any study how the uptake of arsenic in rice plant and their bioavailability into the human body, and maybe possible contribute to the cause of various chronic disease because we consume rice in Indonesia commonly. I think this question addresses to Dr. Tuti. Please, Dr. Tuti, your time to respond the question.
Thank you, Ibu Crescentia, for the question. Yes, the arsenic species in seaweed, mostly in the form of arsenoriboside, or we call it arsenic sugars. Their toxicity, there are many research to see how their toxicity and to look at their metabolic and biochemical properties of the arsenic sugars. But still, it's not the data that we have currently is not enough to make it like the standard from Codex to make it like a standard. The limit of arsenic sugar consumption daily is like this, but everyone agree. All the experts in arsenic agree that there is a potential hazard in there, but how much the hazard we still don't know yet. But it's better to like reduce it. But for some species of what is that, algae or a seaweed, say that we have nori, and then we have kombu, and then we have a kombu from kelp. They have 50% of the arsenic in the form of inorganic arsenic. That's why the most prone people to arsenic contamination from seaweeds is actually Japan, because they eat seaweed like almost in daily life. But if we consume seaweed, so we need to make sure that it's not like too much for daily, to reduce the arsenic contamination. Thank you.
And how about in rice? You still mute?
Yeah, rice is in organic arsenic, so almost all in organic arsenic. Although we still found a metabolic species like DMA, demethylated arsenic, or monomethylated arsenic, but still both methylate arsenic, still both organic arsenic, still potential hazard. But the majority of the arsenic forming rice is in inorganic arsenic.
So do you think that the standard from Indonesian government should be organic arsenic in rice should be
inorganic
stated?
arsenic? Yeah. It should be stated in inorganic arsenic, because there is no standard currently from Indonesian government for rice.
Mm-hmm.
But we eat rice almost every time we eat, so it's very important to set the standard for arsenic in rice. Yeah.
I think that the fact of we are eating rice every day should be considered by the
I think. Yeah, if you want to know, Rose, in Indonesia, if we're not eating rice, we say we're not eating anything at all. So we say that we eat something if we eat rice.
How much of the rice in Indonesia is imported? Is there rice coming from places like Bangladesh that have substantive arsenic challenges?
Our rice is coming from all over the world now. Before we have plenty of rice production in Indonesia, but now we also import rice, and from Bangladesh, India, which contain high arsenic in their rice. Yeah, I think rice in Australia also, because the plantation of the rice field in Australia, it's in the middle part, also have arsenic contamination.
The issue in Australian rice, and someone actually asked the question earlier on about biotoxin contamination and irrigation, is that the selection of selection of rice crops that don't need much water. So we're selecting the types of rice that are very water efficient. Yes. There's also generated a problem that they accumulate arsenic more strongly. So in order to try and deal with water shortage, we've selected for particular types of rice, and those types of rice are accumulating arsenic at high levels. So there's some trade-offs there between irrigation and biotoxin contamination.
Yeah, that's right. Yeah, I think in some of the research say that they try to find modify the genetic of the rice, so it doesn't need much water, because the arsenic coming from the watery from the rice. So if you see that the difference between wet and rice, wet very low arsenic contamination, but rice, because of the iron plaque in the root of the rice, that's like attracting arsenic, and then take it into the metabolic system of the rice.
Okay, thank you, Dr. Uti, for your response. I think there is another two question from Dr. Adi and Dr. Gufrani addressed to Professor Thompson. The first question is, how does seasonality affect the risk of biotoxin contamination in freshwater resources used for irrigation, especially for irrigation for food crops? And the second question is what is the practical statement about the threshold to maintain sustainable water in the river, especially used for agriculture? Does it will modify the increase of photosynthesis or carbon capture and organic matter? So time
Yeah.
for you, Professor Thompson, to respond that to question.
The seasonality question is a really interesting one. Actually, seasonality is much less important in terms of contamination than year-to-year variability is. So in very dry years, when we use irrigation, it brings contaminants that are in the soils to the surface. So we have an issue with salt in particular, but associated with that salt is a relatively large heavy metal load. So if we've had a series of dry seasons where we're irrigating, so water wouldn't naturally be on the floodplain, but we're moving water onto the floodplain to irrigate, that increases the contamination load. And so the concern in Australia is that if we move to a climate which is characterized by long dry periods and then very wet periods, we'll store water in dams, and then when we use it for irrigation, we'll increase the contamination load in the driest years. So it's probably less about season and more about drought years and non-drought years. This question around what's the threshold to maintain sustainable water? That's a question that society has to decide on. So you have to say, what are the values you're trying to protect in your river systems? And if it's large-bodied fish or if it's coastal fisheries, then you need to work out how much water you need to leave in the river to protect those other values. In the sort of systems that Indonesia has, which are characterized by floodplain watering, so naturally floods would have come up onto the floodplain, and that's what's supporting those agricultural areas along the edges of rivers. Clearly maintaining some frequency of flooding so that you are replenishing the carbon on the floodplain is really important. And it's a real tension through most of Southeast Asia that as the demand for land increases and populations increase, we push people down into these floodplain areas. We then build floodplains to protect housing, but the farming areas then are not flooded regularly enough to support the carbon on the floodplain, and the floodplain carbon goes down and productivity goes down because we export more carbon down the river. So again, there's some interesting trade-offs there between demands for different types of land use. But yeah, the real answer to your question is that society needs to decide what environmental values it wants to protect, and then that determines how much water you need to leave. And that's, again, goes back to the earlier point, you need members of society who are able to help make those decisions, who know enough to make those decisions in a sort of informed way.
Okay, I think that's all the answer cover the question from both Adi and Dr. Adi and Dr. Gufrani. Now we are come to the end of the presentation session. So thank you, Professor Thompson and Dr. Tuti, for your valuable insight and for answering our question and doing discussion. I would also like to thank to all participants here joining us online through Zoom and YouTube today. And in conclusion to the webinar, we can highlight that the critical issue of chemical hazard transferring from the environment to the food cycle. So from the discussion of weaving ecosystem function and indigenous values into agricultural landscape in Australia, we have gained deeper understanding of the complex balance between environmental issue and how to manage agricultural production alongside biodiversity, carbon management, and cultural values. So moreover, from the second presentation about arsenic in bioaccumulated in food especially, so that the understanding the pathway and the impact of this contaminant is essential for developing effective strategy to mitigate their presence in our food supply, including making the regulation. It is also crucial to enforce strong regulation and continuous monitoring for particular food that have high arsenic, including in inorganic or maybe total arsenic level. So it should be safe within the safe limit, the concentration. So by doing so, we can protect both not only marine ecosystem but also human health. So ensuring that the water, especially, continues to be safe and sustainable source for food, for future generation. So I think that's all from this presentation today that can be as a take-home message. Thank you for the attention and participation again, and I will now hand it back to our host, Mr. Yuri, who will guide us through the next part of our webinar. Thank you. Thank you for all.
Okay, thank you, Dr. Rades, for your as the moderator in the webinar today, and thank you very much also for our speakers, wonderful speakers for the webinar today. And ladies and gentlemen, finally we come to the end of this webinar, and would like to award certificates of appreciation to the speakers. And please to the host to display the certificate. Yeah, so this is the electronic certificate for the Prof. Ross Thompson. Thank you very much for your today, the webinar, as the and then the certificate for the Dr. Tuti, please, the co-host. Would you? Yes. This is the certificate for the Dr. Tuti Hartati as the speakers today. Okay, thank you, sir. And thank you very much for your valuable contribution today. And we will send to your email soon. And ladies and gentlemen, it is the end of our webinar, and we would like to thank you for spending your time with us today and all the active participations. Thank you for the speakers, moderator, and the committee, and the last but not least to the participants. And ladies and gentlemen, and we would like to apologize for the inconvenient matters during the webinars. Thank you very much, and see you in the next webinar, and goodbye. Thank you.
Thank you, Pak Yuri.
Okay.
Thank you, Pak Yuri and
Thank
team.
you.
And
Thank
thank
you.
you,
Thank you. Thank you,
Dr.
Ross.
Tuti and
Thompson.
Professor Ross.
See you again.
Thank you.
I hope we
Yeah.
can see you in Indonesia later on.
I do too. I'd love to be there too.
Okay, thank you, everyone. See you.
Research is our strength to overcome any limitations. Innovation is the guiding vision that drives us towards the future. We cannot face this alone. We must stand together to make our vision a reality. Growing strong upon the land of Nusantara, a magical place filled with miracle and wonder. A land spanning 2 continents and 2 oceans, rich in culture and biodiversity. we and Indonesia are the necessary combination to realize the grand visions of the future. Like an orchestra, BRIN harmonizes research, development, assessment, application, and also synergies, invention, and innovation in Indonesia. We created an ecosystem consisting of 7 deputies, 12 research organizations, and 85 research centers dispersed throughout Indonesia. Research Organization for Aeronautics and Space, Research Organization for Nuclear Energy, Research Organization for Energy and Manufacture, Research Organization for Earth Sciences and Maritime, Research Organization for Life Sciences and Environment, Research Organization for Electronics and Informatics, Research Organization for Social Sciences and Humanities, Research Organization for Archaeology, Language, and Literature, Research Organization for Health, Research Organization for Nanotechnology and Material, Research Organization for Agriculture and Food, Research Organization for Governance, Economy, and Community Welfare. BRIN catalyzing research and innovation to develop an advanced scientific ecosystem in Indonesia with 2 main targets. BRIN realizes the tremendous potential of the nation's youth in the field of science and technology. Thus, various research and innovation policies are being formulated to develop and cultivate human resources with high competence and aptitude in science and technology. Creating many excellent programs is a form of BRIN's commitment to researchers, innovators, and the general population from various disciplines across the nation. The research and innovation ecosystem quality improvement in Indonesia is also realized through the collaboration of various ministries, institutions, domestic industries, and regional governments, to name a few, by forming the Regional Research and Innovation Agency, or RIDA. Moreover, cooperation with various foreign research institutions continue to be developed through active participation as member of various organizations and international forums. BRIN organizes research into a more effective and efficient process by integrating human resources, infrastructure, and budget from various research and development institutions. Oriented by a national research priority, BRIN routinely enacts comprehensive evaluations from various research and development institutions. This effort is supported through the continued management and improvement of various research and innovation facilities located across 17 regions of Indonesia, dedicated towards the development of science and technology. BRIN continues to improve our contribution to research and innovation towards national development by developing and revitalizing 4 areas of science and technology, or KST. An integrated area consisting of research laboratory facilities, testing laboratories, and industrial facilities for both startups and SMEs, tenant office rooms, co-working space, meeting rooms, gallery rooms, and other supporting facilities. BRIN is here to manifest the future of a golden Indonesia 2045. Like consolidating science and technology resources and creating a resource ecosystem and research-based economical foundation. Transforming Indonesia as a global reference in the development and application of science and technology, practices, and the good of humankind. Nature is the source of inspiration. Knowledge is the foundation, and to be put in praxis for the sake of the nation.